Hello everyone and welcome to today's webinar, Deep Learning in Optics. My name is Laurent Bentolila from the California NanoSystems Institute at UCLA, where we perform some of the work which is going to be presented today, and I'll be your moderator for today's event. Today's educational webinar is presented by LabRoots and brought to you by Leica Microsystems. To learn more, please visit Leica-microsystems.com. So let's get started. You can pose questions to the speaker during the presentation while they are fresh in your mind. To do so, please simply tap them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. To enlarge a side window, click on the arrows at the top right-hand corner of the presentation window. If you do experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Ask a Question box located at the far left of your screen. This is an educational webinar which offers free continuing education credits, so please click on the Continuing Education Credit tab located at the top right of the presentation window and follow the process of obtaining your credits. I now present today's speakers, Professor Aydogan Ozcan, Professor Gary Riverson, and graduate student Hongda Wang from the Electrical Engineering Department at UCLA. For a complete biography of our speakers, please visit the Biography tab at the top of your screen. Professor Ozcan, you may now begin your presentation.
Thank you. Thanks, Laurent. So hello, everyone. It's my great pleasure to introduce our team and our work on deep learning in optics to you. So basically, this is going to be a presentation given by three of us, myself, our graduate student Hongda Wang, and our junior faculty Gary Riverson. So before I dive into the topic of today, I'd like to give you some brief introduction to our lab. We work on computational imaging and sensing with applications in biomedical space, point-of-care diagnostics, mobile health, telemedicine, and more recently, environmental monitoring. A key element in our research has been the use of smartphones and smartphone components for essentially creating mobile and cost-effective interfaces. This line of research has come a long way. Maybe more than 10 years ago, with a smartphone-based microscope, we could barely see red blood cells, but these days, actually, you can utilize a mobile phone-based microscope in a cost-effective manner to even see individual DNA molecules and size them with up to about a kilobase pair sizing accuracy. As shown here with a simple, inexpensive interface, you can see fluorescently tagged DNA molecules and size them with about 1 kilobase sizing accuracy. And this is actually quite relevant for bringing genetic testing to these types of microscopes. In a recent work, we've performed targeted DNA sequencing and in situ mutation analysis using a multimodal mobile phone-based microscope, which had bright field in addition to 2 different channels of fluorescence, where each fluorescent channel was used to look into amplified signals coming from tissue to reveal mutant and wild-type genetic sequencing happening through rolling circle amplification, a process that amplifies DNA from a template and essentially creates a cluster of DNA that's labeled and imaged by the smartphone-based microscope. These are some highlights about how we can utilize smartphones and smartphone-based components for microscopy and for sensing. And in this slide, I'm sharing with you some of the examples from our lab for the utilization of the smartphone as a sensor for waterborne pathogens, for heavy metals, quantification of different kinds of diagnostic tests. These are now enabled through smartphone-based interfaces. And in a nutshell, essentially, this line of research is aiming to democratize advanced measurement tools by making them cost-effective and compact, while at the same time providing the same level of specificity and sensitivity and resolution that you expect from lab-grade instrumentation. After this brief intro, I'd like to come to essentially the topic of today, which is how we're using deep neural nets for image formation, image reconstruction, and image transformation. If you look at today's symbiotic relationship between professionals and machine learning, we see some very interesting and symbiotic relationship in the sense that especially machine learning techniques are being used at the front end to help diagnosticians see through many different images using statistical learning. So helping essentially diagnosticians and professionals to deal with an exponential growth in the images to be diagnosed. And we've seen the impact of deep learning in this field tremendously. Whether it's actually detection of cancer, detection of tumor margins, grading of tumors, and looking into segmentation and a detection of different kinds of features within different medical images. These are some of the examples of how deep neural nets and deep learning in general have been utilized to help diagnostic analysis of various different medical images. Today, though, I'd like to emphasize another very exciting opportunity on the use of deep neural nets, and that's actually exactly highlighting this new opportunity, which is enhancing the images coming from microscopy tools by improving them, by creating new kinds of image transformations, image reconstruction methods powered by deep learning. And today I'll share with you some of these examples of how we're enriching microscopy experience through deep neural nets. And I'll start, before I pass it to Hongda and Yair to present other results on this, I'd like to give you some examples on how machine learning is helping us in holography, in holographic image reconstruction. In a nutshell, we're migrating from physics-based reconstruction methods to data-driven, image data-driven reconstruction methods. So in this example that I'll share with you, we're going to teach a neural net some physics through image data, help the neural net essentially solve holographic microscopy imaging problem, inverse problem. So in holography, one of the most important things is actually phase retrieval. A hologram encodes the information of the sample in interference patterns, as shown here, for example, and you need phase to reconstruct this sample. Only after phase retrieval you can actually go back to the sample plane and reveal from these holograms shown here to what you would like to see, the subcellular features of, in this case, a tissue sample. Over the last several decades, various different research groups utilize physical insights to create different types of phase retrieval and holographic reconstruction methods, oftentimes involving different types of measurements, where you would, for example, change the angle of illumination, wavelength of illumination, or, for example, sample to sensor, to encode more information in your recordings, in your holograms, to be reconstructed physically, to go from a hologram as shown here, for example, into a nice phase reconstruction. So we spent a lot of time as a community to mature this field so that we can reconstruct images without any artifacts or with least artifacts using physical techniques, physical methods. Deep neural nets are now changing this game for us. We like deep neural nets for inverse problems in general because, yes, it takes a while to train them. It can be a few hours to maybe up to a day, even longer sometimes, to train a neural net. But once it's trained, it's fixed. And it's a single feed forward without any iterations. And this is actually a game changer for us because a lot of physical reconstructions in holography and other inverse problems that the computational imaging field has been tackling, there are always iterations between your guesses and what you want to reconstruct. And this is now changing the game because deep neural nets can actually solve various different optimization problems in a single feed forward very efficiently and rapid. This is essentially what we're using through image data, through holographic image data, to teach the neural net to do this physical transformation. In this example, we're looking at inline holography, where the sample goes in front of a CMOS imager, typically sub-millimeter gap between the sample to sensor, and we record inline holograms of the sample. Only after phase retrieval, you'll be able to see the specimen as shown here. These are pap smear samples that we're trying to reconstruct. In this case, a convolutional neural net has been trained with pairs of images, typically close to 10,000 patches of input and output images are used to train a neural net like this, where at the end, the neural net actually learns to transform a reconstruction that doesn't have any phase information into a reconstruction that has the phase recovered image of the amplitude and phase information of the specimen, which is essentially creating an alternative path to physics-based reconstruction methods. In fact, the neural net was trained with a gold standard physical method to get the ground truth images during its training. This training can take, as I said, more than 12 hours, depending on the type of the sample that you want to reconstruct. But once it's trained, it's actually fairly robust, and it gives you the same kind of reconstruction that you expect from a physical reconstruction. What you see here is a comparison of the network's output in a blind inference. The sample has been never seen by the network, and how it compares to a physical reconstruction. One thing that's very exciting here is that some of the artifacts that are coming from the temporal and spatial coherence of the illumination are mitigated in the network output as well, which is a very unique feature of these network-based solutions, in the sense that it's a transformation from an input holographic field to a cleaned, artifact-free, two-image artifact-free phase retrieve reconstruction. It's a plane-to-plane mapping in this case, and any of those dust particles or other particles are actually washed away at the reconstruction. In the next example, I'm showing you the same type of a result with a tissue sample. This is a breast tissue section where the network input is shown here on the left, the network output is shown in the middle, and a physical reconstruction giving you exactly the same thing with 8 different hologram acquisition is shown on the right, which shows you the success of the neural net in a single inference. In terms of savings, obviously we have also savings in image acquisition, hologram acquisition, and reconstruction time. What you see here is a comparison of a physical reconstruction method which uses multiple heights. For example, if you use 3 different heights, sample to sensor distances, the reconstruction takes about 28 seconds for a certain field of view of about a millimeter square. If you use, for example, 5 heights, 5 different sample to sensor distances, it's about 35 seconds. Our neural net actually is fairly fast, and it's saving time because of the non-iterative nature of its inference. At the same time, it's also reducing the number of image acquisition steps. So next, I'll share with you some additional things that you can teach a neural net to do some more exotic things. In this example, we're going to do not only phase retrieval and holographic image reconstruction, but also do auto-focusing. So the neural net will be trained here by defocused holograms, where the ground truth will be individually focused by a physical autofocusing mechanism. And at the end of this training process, the neural net is actually going to bring various different parts of a 3-dimensional sample into focus, and at the same time phase retrieve, eliminating 2-image and other artifacts of coherent imaging. A good example of this is shown here for imaging a 3D distribution of bioaerosols. These are particles captured from air that have different heights. If you take a physics-based reconstruction, which is shown here in B, as you scan through the sample volume at different heights, you'll be seeing different particles coming into focus. For example, if you move from -10 micron height to +10 micron height, some particles get out of focus, some particles get into focus. Whereas in the network that is trained with the focused holograms, you see that the inference actually brings all of these particles that are 3D distributed into focus at the same time. So we have an extended depth of field within which we can bring individual particles into focus while also retrieving their phase, which is actually quite interesting in the sense that it doesn't satisfy the Maxwell's equations. We extend the depth of field, and this depth of field improvement is also shown in part C. For a single particle, as you move the particle to different heights, you see natural defocusing as a result of the wave spreading. But in the network results, it's actually in focus over plus minus 100 microns, which is the range that it was trained for. The network was trained within plus minus 0.1 millimeter. This also works for tissue samples. It's shown here. This is a connected tissue sample. The neural net works very nicely over 0.1 millimeter in one direction, whereas standard physics-based reconstructions quickly get out of focus as you defocus the sample. So these are not just cool results in the sense that they're helping us provide alternative methods for reconstructing holograms. In fact, we're using them in different kinds of mobile instrumentation. For example, recently we've created an imaging flow cytometer that can look into flowing objects with a beautiful resolution in red, green, and blue and synthesize colorful images of these flowing objects. This is powered by deep neural net-based holographic reconstructions. Another device was looking into bioaerosols and sensing pollen and mold particles without the use of any labels. It's a label-free bioaerosol detection system. This is also powered by the extended depth of field and holographic reconstruction created by deep neural nets. And recently, we've also utilized the same framework for sensing of viruses using lensless holographic microscopy techniques. In other words, these interventions, these kinds of ideas are helping us enable high-throughput screening, high-throughput imaging sensing devices to look at extremely large volumes of samples, air, water, or bodily fluids, even in field settings. And that's where we're utilizing neural nets to get these mobile devices even more powerful. What's very exciting about this framework is that it can actually also transform images from one modality to another one. This is something that Hongda and Yeri will emphasize more with various other biomedical applications. But I'll finish off my part of the presentation by telling you how you can actually train a neural net to take a holographic reconstruction and transform it into an image that's the color and image contrast of bright field microscopy in terms of the transformation. Here, we're training a neural net based on holograms, and the ground truth is coming from a scanning bright field optical microscope. So we're actually having best of both worlds, 3D scanning capability of holography digitally, and the beautiful color and image contrast of incoherent bright field microscopy. And the results are summarized here for some examples. These are pollen particles. Holograms are shown here. If you backpropagate these holograms to different heights, you see artifacts of coherence, just like an inline holography. The neural net takes these and reconstructs the same color and spatial contrast, a feature contrast of bright field microscopy as if it was scanned in 3D with a bright field optical microscope, eliminating all these artifacts, but also bringing the contrast mechanism of bright field microscopy. And this is obviously very advantageous because we can take a hologram, backpropagate it digitally to different heights, and at each height, get the corresponding bright field image coming from a high-NA objective lens, all through virtual transformations enabled by deep learning. And this is actually indeed quite powerful in what it can achieve. And in the next part of this presentation, Hongda is going to talk to you about how these cross-modality image transformations help us achieve super resolution, help us, for example, transform a confocal microscopy image into STED microscopy equivalent images and many other possibilities. So now I'll pass it to Hongda for this part of the presentation. Hongda?
Thank you. I'm Hongda Wang. I'm a graduate student at UCLA. So next, I'm going to share with you our recent progress in cross-modality image transformation that achieves super resolution in fluorescence microscopy. So to begin with, we first show you three major super resolution techniques. These are stimulated emission depletion microscopy, structured illumination microscopy, and localization microscopy. So these techniques provide unprecedented access to the inner working of cells and various biological processes. However, these techniques often rely on sophisticated optical setups and extensive computational processing of the image data. In fact, in one of Dr. Basic's talks, there is a summary of the current techniques. So in order to acquire super resolved images, the STAD and the localization microscopy techniques require much more photons, somewhere between hundreds to hundreds of thousand-fold. The structured illumination microscopy does not require that much photons, but needs multiple exposures and computational reconstruction. So here we present a deep learning-based framework to achieve super resolution and cross-modality image transformation in fluorescence microscopy. We demonstrate a possible method for high-throughput microscopy and the potential to democratize super-resolution microscopy to more researchers. In our method, by training a neural network model, we can transform a low-resolution image into a high-resolution one. Our method is purely data-driven, which means it does not rely on any prior knowledge or modeling of the imaging process. Furthermore, we also show that the neural network inferred image has extended depth of focus and improved signal-to-noise ratio. So how do we train a neural network? First, we start with the generator network model. It takes a low-resolution image and enhances the resolution. We then compare this network-enhanced image against the high-resolution image acquired by another modality and calculate a pixel-wise loss. In this work, we used mean square error and structure similarity index. This is the first part of our network loss. Then we have another discriminator network model that tries to distinguish the network-enhanced image and the true ground truth image and gives us an adversarial loss. This is the second part of the loss. So during the network training, the generator tries to fool this discriminator network, and the discriminator tries to distinguish the two images. When they are in an equilibrium state, the network is converged. So we have a generator model trained that can take the low-resolution image and enhance it to match the resolution of high-resolution image. We have to emphasize that the accurate registration of the low-resolution and high-resolution images is critical to the performance of our network model. So we first demonstrate the resolution improvement with wide-field fluorescence images. You can see here on the left is the network input image captured with a 10x objective lens. On the right, you can see the ground truth images that is captured with 20x objectives. The green-colored microtubules and the magenta-colored effectins are known as cytoskeleton filaments that keeps the cell shape. The network output images reveal the fine structures and agree very well with the ground truth images. We also noticed that our network inferred image has extended depth of field. We can see in this slide, here's a mouse blood vessel that grows out of the plane. If you look at the network output, although it's a 3D structure, everything is in focus. This is highlighted by the green and the yellow arrows pointed regions. We can further confirm this effect with an extended depth of field image that is synthesized from a Z-stack of 34 images. We can also apply this framework to confocal images and super resolve them to match the resolution of STAD microscopy. Here, these images show 20 nanometer fluorescence nanoparticles. some of them are closely spaced that cannot be resolved by the confocal microscope. However, the neural network can successfully resolve them and matches the ground truth images. We also draw cross-sections to confirm this agreement. As a commonly used method to quantify the resolution in super-resolution microscopy, we characterize the point spread functions. Here we selected more than 400 individual nanoparticles at the same locations in confocal, STAD, and network inferenced images. We fit the individual nanoparticles with Gaussian profiles and calculated their full-width half maximum distributions. As shown on the right side, the full-width half maximum of confocal microscope is centered at around 290 nanometers, roughly corresponding to a diffraction-limited imaging system, while the full-width half maximum distribution of the network output images and the STAD images are at around 110 nanometers. This beautiful shift of PSF distribution clearly indicates the resolution improvement. Furthermore, the network output image also has improved signal-to-noise ratio compared to the input and the ground truth image, as shown by an example of confocal to STAD image transformation here. And we actually believe this is one of the major reasons that the network can deconvolve the input image with higher signal-to-noise ratio. We can also estimate the local point spread functions. You can see that we deconvolve the network output image with regarding to the network input image and show the PSF of local regions. We repeat the same operations with ground truth image and input images. We see that the network can actually learn the spatially varying point spread functions and perform an adaptive deconvolution to super resolve the nanoparticles. So next, we can also apply this confocal to STAD image transformation framework to super resolve histone 3 distributions within fixed HeLa cell nuclei. You can see here the network input, output, and ground truth images shows that the network successfully improved the resolution of a confocal microscope image matching the STAD image of the same nuclei. We draw the green and the yellow cross-section profiles to confirm this agreement. Next, we demonstrate our cross-modality image transformation framework by transforming diffraction-limited TURF images to match the TURF SIM reconstruction. TURF means total internal reflection fluorescence microscopy, while TURF SIM means TURF with structured illumination. This thing on the left is called clustering-coated pit. It is one of the mechanisms that the cell bring different cargoes inside the cells. So they form these dome shapes inside the plasma membrane, and the cargo sits inside the domes. When we are doing TURF, we only illuminate the parts that are very close to the plasma, so they appear as rings. These clustering-coated pits have their own dynamics. For example, they associate with actin, and they also spin off pits. However, these structures are so small that cannot be resolved by diffraction-limited methods, as you can see here in the TURF images as the network input. We imaged the SUM159 cells that expressing GSP-labeled clustering adapter AP2. To highlight the examples, the neural networks was able to detect the deassociation of clustering-coated pits from larger clustering plaques. As shown on the bottom row, it can also detect development of culture-bearing clustering cages, as shown in the middle row. There are 3 cages here. In fact, this framework is so robust that we actually applied it to a video of the clustering dynamics, and these are the significant frames that we cropped from the videos. So a common concern of computational approaches to enhance image resolution is the potential spatial artifacts. Here we used Nano-Squirrel plugin which is a toolbox to quantify the potential artifacts for super-resolution microscopy between a low-resolution image and a super-resolved image. We calculated the error between network output image against the input low-resolution image, as shown here. The resolution-scaled Pearson correlation is almost 1, and the resolution-scaled error is 0.9 out of 255, which means a high degree of agreement of the network-enhanced image and the network input image. To understand the scale of this error, we also calculated the ground truth image against the network input image. In fact, the ground truth image has larger error and lower Pearson correlation coefficient. We think this is partially from the limited depth of field of the high NA objective. To do a more intuitive comparison, we also overlaid the network-enhanced image and the ground truth image in different color channels. If there is any mismatch or artifacts, we should be able to see a color block there. However, we see that it's a uniform white-colored image that indicates the high degree of agreement. Another way to look at this result is to do a spatial frequency analysis. Here we look at the spatial frequency spectrums. The cross-sections of the radially averaged power spectrum confirms the success of the network output, matching the extended spatial frequency spectrum from a higher resolution imaging system. So conceptually, the network is performing a knowledge-based frequency spectrum extrapolation. Next, we demonstrate the generalization ability of our network to enhance the image resolution of objects that it has never seen before. On the left, we see a network model that is trained with microtubules of a cell and applied to F-actin melanoma cells and the mouse kidney. It super resolves them and match very well with the ground truth images. On the right is a network model that is trained with F-actin images and then applied to mitochondria, blood vessel, and mouse kidney. They also match very well with the ground truth images. In fact, the extended depth of field images we showed you previously is a mouse blood vessel, and it's a new kind of sample that the network has never seen before. It still generates a all-in-focus, high-resolution image. We also test this generalization capability with our turf-to-turf SIM transformation framework. Our network model was trained with SUM159 cells, and here we applied to Drosophila embryo tissues. It clearly resolves these fluorescence particles and agrees very well with the ground truth images. These frames are also cropped from a video that shows very robust framework capability. However, we have to emphasize that whenever possible, an optimal model should be trained for new imaging modalities. Here we show that by using a model trained with Texas red filter and F-actin images, it can improve the resolution of the turf images and confocal images. However, the image quality cannot match the optimal model inference. This does not mean the network does not have generalization capabilities. It still improves images, and it's a further question to quantify how far we can push an existing network model to infer new images. So in summary, we presented a deep learning-based cross-modality super-resolution framework that takes a single image as input and transforms it without iterative process. The success of our method is demonstrated by low to high NA wide field microscopic image transformation, confocal to steady microscopic image transformation, and turf-to-turf SIM microscopic image transformation. This purely data-driven approach does not need any prior knowledge of the image transformation model. We also showed that the network inferred image has extended depth of field and improved signal-to-noise ratio. And at last, we show that a pre-trained network model can generalize to new types of samples it has never seen before.
So here we demonstrated cross-modality transformation from a low-resolution fluorescence image to a high-resolution fluorescence image taken by a more sophisticated setup. Next, we will also demonstrate cross-modality transformation from auto-fluorescence images to the equivalence of bright field images, which will be brought to you by Dr. Rivenson. Thank you.
Thank you, Hongda. Hi, my name is Yair Rivenson. I'm a junior faculty in the Electrical Computer Engineering Department in UCLA. Now we're going to talk about how deep learning can do a very interesting task or even translational task of virtual staining of label-free tissue sections for histopathology. In a nutshell, histopathology is the diagnosis and study of diseases of the tissues. Since decades and hundreds of years ago, it's been used to look at cells of tissue and tissue structures under a microscope. The basic process goes as follows. A patient will, after some sort of screening process, will undergo a biopsy, and then that biopsy will be fixed and sectioned into small tissues, which will be examined under the microscope. Unfortunately, if you would look at the tissues as they are in their native form under a bright field microscope, you'll see a very poor contrast, which doesn't provide any clinical information. At that point, that's about 150 years ago or so, scholars have devised all kinds of techniques of staining the tissues, basically utilizing the chemical environment, the subcellular chemical environment, to bind specific chromophores to the tissue sections and provide a contrast that can be used for diagnosis, which since then has been the gold standard in numerous types of diseases, and specifically cancer. So histochemical staining has a few drawbacks. First of all, it's a laborious process. It is time-consuming, especially if you go to more of a special stain. It's an expensive process which requires multiple chemical reagents. It requires training, specific personnel, histotechnologists that are doing the staining. It requires a lab space. Some of those stains requires monitoring by the histotechnologist. The other thing, it doesn't support tissue preservation for advanced diagnosis. And as you can see here, this process is prone to staining variation. So basically, different technicians in the same lab and between labs might provide different staining results, which makes it harder for a diagnosis for different pathologists. And today, a growing number of machine learning algorithms that use those scanned tissue samples images as inputs. So about, let's say, 2 decades ago, there has been a substantial effort to come up with alternative contrasting methods. And there were basically 2 schools of thought. One of them is rapid staining. Rapid staining basically takes a tissue, a fresh tissue, or a section tissue, and put it into some staining chemical bath, which, for example, might be acridine orange or eosine, and then image it with some sort of an alternative microscopy technique, for example, two-photon or second harmonic generation or UV surface excitation. The other school of thought went with completely label-free imaging and then acquire multiple images at multiple modalities. Could be multi-photon or high harmonic generations. Another very popular approach is Raman microscopy and also quantitative phase microscopy more recently. So all these provide you with some contrast, which can be translated into diagnosis. However, most professionals, clinicians, are not trained with this kind of imaging modalities. Therefore, there needs to be some sort of a bridge between those methods and what clinicians are used to look at, because retraining the expert and also algorithms that today are well trained with millions maybe of images coming from standardly stained, histochemically stained tissues, will also be very costly, and also there is almost no data for any other modality out there. So the solution would be what's called pseudo staining, which was based on taking basically the contrast that's created by one of the methods that we described, and do some approximation to the intensity of the dye concentration at that specific pixel or group of pixels. And it will require multiple images or spectra or some sort of a rapid staining mechanism that we also mentioned before. So here we demonstrated a deep learning approach that is completely label-free, which is we call it virtual staining, and it's using basically a single auto-fluorescence image of the section tissue, and it has the potential to eliminate the histological staining process, and from a single auto-fluorescence image to create an image a matching image from a bright field microscope following the staining process of the tissue. So for that, we chose, for the autofluorescence wavelengths, we chose the DAPI because it has a good excitation profile for many constituents in the tissue. So for the training, we used, again, a generative adversarial network, as Hongda mentioned a few minutes before, which takes the autofluorescence image and tries to fool a critique network or a discriminator, and that balance between the networks generates a high-quality image that matches the stained image that we had seen in the ground truth. Again, it's also very important to mention that we're doing a multiple stage registration and we saw it's extremely important to do this stage because to get the highest quality of images between the result of the network and the matching ground truth and sort of preserving all the features of the tissue. In terms of training time, it varies, again, depending on the number of data samples that we've had. Some of it went all the way to 2 days. But again, it was trained on standard desktop machines, and obviously this is scalable with higher performance servers. Once this thing is trained, it can output an image at about the times that we calculate is about 1.9 seconds per millimeter square, which stands in line with the scanning speed of traditional microscopes, or even faster slide scanning microscopes. So once we trained the network, here are some examples of results. First of all, we did the standard HHMI staining, which is the most basic staining. About 85% or so of the stains that are being used today are HHMI. And the annotations here that you see with the errors and the stars were made by the pathologists that we worked with us on this research, showing some features that that were important for the diagnosis of this tissue. This was for the salivary gland, but we did it for other tissues as well. So what is extremely special about this framework, this was the first demonstration of special stains. Here is an example of a Mason trichrome stain for a lung tissue. And again, till now, there has been no demonstration of anything else other than HHMI, which is pretty simple to perform, even with pseudo staining techniques. And the other stain we've done here is what we call the Jones-Silver stain. This is for the kidney tissue, and this is a basement membrane stain that is used many times in a diagnosis of diabetes. In this case, there is a tumor on those tissues, but we still use the silver stain. The most important thing in here is to beyond our analysis is the blind assessment was done by the pathologist. At the end of the day, we gave those images to pathologists and blindly assigned them either virtual or histological stains, and they had to perform some diagnosis based on the images that they have received. And the conclusion of this study, which was again performed by board-certified pathologists, that there was no major discordance or no clinically significant difference in the diagnosis between the virtual and histological stain. And the other assessment that the pathologists have done was what we call the staining efficacy. And the staining efficacy or stain quality, we asked them to give a grade between 1 and 3, and two different cytometric details that we asked them, and the overall staining quality. And we were very happy to learn that there was, again, no specific preference to either the virtual or histochemical stain. In fact, most of the pathologists liked the virtual stain more than the histochemical stain. One of the interesting attributes of this framework is that it standardized the staining. If you look at the top row, this is a staining of a liver tissue section, and you see there is some staining variation, but the network does not do this staining variation. It converges to some sort of a common color palette or colorization panel of this tissue section, and it will be very helpful for both clinicians and machine learning algorithms in terms of this staining standardization. Another interesting thing was that, for example, when we looked at the sample that contains melanin, and melanin basically absorbs almost all the light at the illumination wavelength that we use, the DAPI. And so as we can see here on the left image, when the network tried to reconstruct that, it basically thought the melanin is part of the background. But when we added a low resolution scan with another wavelength, again, everything is auto-fluorescence label free, we were able to reconstruct faithfully that melanin. So it has emerged, and this is one of the ways for us to try to add more information, if necessary, to this virtual staining framework. So to summarize, this is another example of how deep neural network can create interesting opportunity for microscopy. In this case, it transforms a label-free image of a section into how it would look into a stained version using a standard microscope and filter set. It's very compatible for low resource settings, and it supports to do smaller biopsies. It can do multiple stains on the same tissue section. It can handle multiple fixations, such as frozen sections and paraffin fixation. It can accommodate other modalities, such as quantitative phase microscopy, which we have demonstrated in another effort. And it can enable more advanced analysis after we have done the virtual staining, because we're label-free, and then we can just send it to more advanced analysis. So this concludes our talk. Thank you for listening, and we're looking forward to getting some of your questions.
I'd like to thank our three speakers for their very informative presentation. So we will now start the live Q&A portion of the webinar. So if you have a question you'd like to ask, please do so now. Just click on the answer and question box located at the far left of your screen. We'll answer as many questions as we have time for. So let's get started. So our first question, actually, we've got quite a few recurring questions concerning actually the concerns that might arise when it comes to artifacts that might be induced by this deep learning network. So the question is essentially, can you explain the different steps that you are taking to ensure the integrity of the deep learning output?
So, yeah, I think that's a great question. And we frequently get this asked. The paper that we published at the end of 2018 in Nature Methods has actually a specific subsection dedicated to artifact analysis. And in the manuscript and the supplementary information, we've given various different methods analysis to quantify artifacts by looking into resolution, by looking at cross-sections of input-output ground truth, by looking at peak-to-peak separation of particles or features within the specimen, by overlaying ground truth against the network inference results at different colors, looking for color artifacts, by looking into spatial frequency analysis, spatial frequencies of the input, ground truth, and the output of the network, and other tools that we've quantified to look at potential artifacts and to verify that indeed we're getting what we would like to match the ground truth. In fact, in some cases, even surpass some of the artifacts of the ground truth. Some of the limitations of the ground truth are also surpassed. And I think Hongda gave some great examples of this with some of the slides that he shared today, in terms of depth of field enhancement, in terms of some of the artifacts that are cleaned up. We've taken this very rigorously, and I think our papers have ample evidence of the fact that what we infer is a very nicely conditional. It's a generative adversarial network, but at the same time, because of how we train it, is conditioning it to infer within the limits of the physical system. And the fact that the input images are also coming from a microscope that is imaging the same sample with some resolution to be enhanced is helping us to essentially retain artifacts within a well-conditioned limit. In the case of the virtual staining case, which also uses a generative adversarial network, we've done similar analysis at the network output to look into spatial frequencies, to look into resolution, to look into structural similarity index, to look into color distance of the network's output with respect to the bright field, actually stochastically stained samples, and shown that they're actually matching each other. But more than that, we've given them to board-certified pathologists for them to look into stain quality, for them to look into diagnostic value of our images. In fact, to the level where board-certified pathologists, a group of them, could not tell the difference between our images and theirs, especially for a specific application like this, like virtual staining in histochemistry, histology as a field. The ground truth is obviously what diagnosticians are accustomed to seeing. And that's why we're confident that what we generate is satisfying the needs of the application, and in that case, the needs of the pathology field, as certified by these diagnosticians and group of them. Was there any other analysis tools that you've used that I didn't mention?
No. Oh, the Squirrel plugin.
Yeah.
So in addition to that, we also used Nano-Squirrel plugin, as I talked in the slide, to calculate the potential artifacts between the super resolved image and the input image. And we show that the mismatch is lower, even lower than the mismatch between the ground truth and input image. So the artifact there is very minimal.
So in fact, a related question that we sometimes get asked is, we're training using ground truth images that have limited depth of field, sometimes that have artifacts. So how can you actually surpass that? That's actually a very interesting question. The input images have extended depth of field because they're captured by a low numerical aperture system. By definition, they have a longer depth of field. So we essentially generalize to bring a larger depth of field into focus with enhanced resolution compared to the ground truth. And that's actually validated in our results by taking our inference results, comparing it to a high-NA image, and we claim some parts of our images in network inference results show features that are out of focus in the ground truth. To show that this is physically the case, we take the same sample and image it with an axial scanning high-NA objective lens and create an extended depth of field by, for example, capturing 20 images with a half a micron or less than that axial scanning to indeed show and validate that the enhancement in the depth of field is a physical property of the input image, which is enhanced in resolution across a larger depth of field. So those extended depth of field images are also part of our analysis as part of these papers to show the advantages of the network inference. Similar analysis also applies to structural illumination microscopy SIM results, which, depending on the aberration, depending on the sample, depending on the field of view, sometimes have certain aberrations, reconstruction artifacts appearing. Those are also attacked by our network. And we have provided evidence of that in our papers and in some of the movies that we have as supplementary information to the paper.
Thank you very much. A few of our listeners are asking how many images are needed for training the deep network.
I'll take it. So yeah, it basically, it depends on the application. For one application that I think was our first paper, deep learning microscopy, we trained with like 10,000 very small patches, 60 by 60 pixels, and transforming them into 150 by 150 pixels. So basically, it's just one slide of a lung tissue section that we scanned and used for training. And with that tissue, with that network model trained from that tissue, we basically were able to infer many other types of tissues and also a resolution, a test target. So and got the highest, higher resolution enhancement as we've demonstrated for just the lung tissue section. In other cases, we might need more data, for example, for the virtual staining. At one case, we had to scan about 6, 7 tissue sections to get that sort of standardized stain. But that thing, again, kind of varies according to your application. But I would say that in general, you need much less samples than you would need for classification frameworks, because here we're basically trying to predict a pixel value or a group of pixels from another group of pixels. So we have in each slide, we already have millions of those. So it's in some senses it's slightly easier task, and we need overall less data.
Thank you. We have one question from the audience here asking if these deep learning models are available to the research community.
Yes, that's a great question. We have made some of our models publicly available. These are also mentioned in the code availability parts of our manuscripts. So we've got also some sample images there, some test images, some comparison ground truth images for them to play around with our models. Later this year, if we have some resources and time, we would like to also open up some of our databases for research community to also work on their own models, their own model development, testing, training processes. So we're also eager to kind of combine some of the work that we've done into a single portal to share some of our raw images, because these are quite exciting, valuable results that we'd like to have other people repeat, expand, and use in their own applications.
Thank you. So it looks like we have time for maybe one more question. So maybe I'll take that opportunity to ask you guys, is there any limits to these approaches? I mean, we have seen cross-modality transformations. We have seen application to histology and pathology. I mean, what else?
So this is a great question. I think Yair and Hongda should chip in. The ultimate limit for these frameworks is signal-to-noise ratio. So, and that's actually also a strength and also a weakness of this framework, in the sense that as the noise statistics change, you will have to retrain, which means as, for example, your microscope changes, it's a good idea. If your type of sample changes, it's a good idea to maybe take your model and train it with the combination of the new types of samples, new types of microscopes that you have. But ultimately, the deep neural net is learning spatial, spectral features of objects, types of objects, to generalize those features that are learned inherent to the network to infer enhanced images. While doing so, it learns and rejects noise. So it's ultimately limited by signal-to-noise ratio. In fact, all microscopy modalities, regardless of how they work, whether they claim to beat diffraction limit, whether they claim to match diffraction limit, or any others, their capability of what they claim and how successful they are with their claims is fundamentally limited with signal-to-noise ratio. This applies to SET, this applies to Palm, this applies to Storm, any other microscopy modality. In this regard, we're on the same ballpark. It's SNR limited. As a result, if you want to jump from optical microscopy to electron microscopy, the noise that we have in an optical microscope at the spatial features that are of interest to electron microscopy won't let you jump faithfully. We've tried that. We've looked into those kinds of transformations from optics to electron microscopy, but we're ultimately limited with what you can assume at the noise level and what noise gives us. However, it's certainly possible to have roughly, empirically, I can say, 2 to 3-fold increase in each direction, for example, for resolution enhancement. The same applies, for example, for virtual staining. If your autofluorescence images go weaker and weaker for certain sub-tissue, subcellular components, then it won't work for us to stain virtually. That's where maybe we should switch to another band of fluorescence where it's better SNR, or maybe ultimately switch to another contrast mechanism, like, for example, quantitative phase imaging, QPI, holography, to generate those label-free images and then switch to virtual staining. In fact, in recent work, we've done that. We've used another contrast mechanism like quantitative phase imaging to do virtual staining. It has certain advantages and disadvantages compared to autofluorescence, but these are essentially at our disposal as weapons, so to speak, that we can utilize to handle SNR challenges, contrast challenges, artifacts that different contrast mechanisms have, to mitigate them, to do whatever we want the neural net to do. It's sometimes also outside the physical world in terms of how we are thinking. It can do transformations that are not possible with standard forward models. And that's actually a very powerful aspect of deep neural nets and how they can help us with image transformations. One great example of this actually is between autofluorescence and bright field imaging with the virtual staining, apple to orange transformation. There's no physical model that you can faithfully construct between the two. A similar conclusion also applies between holography, holographic image contrast, and bright field. One of them is a single frequency, a single color. The other one is actually a white light type of an imaging system. So these are very exciting aspects, and ultimately, in different modalities, different contrast mechanisms will enjoy what deep neural nets can offer to enhance our experience of microscopic images that are going to be feeding into professional diagnosticians for their own use in different settings.
Thank you. Before we conclude, I would like to thank our speakers, Professor Ozcan, Professor Verwesen, and Dr. Wang. And I would like to ask you if you have any final comments for our audience.
So one final comment that I would like to add is, let's say we have a model that we trained with our microscopes, but and we already experimented with data that was generated from other microscopes and used the concept of transfer learning, basically either adding one more layer to the deep network or do some sort of a soft restart of the training of the network from the weight it was already trained with, but very small amount of data. And then we were able basically to generalize to the new data, to the new task that we've got to enhance. And so far from some of the examples that we have done, it worked quite nicely, again, with a very limited number of images.
Thank you again, Professor Aydogan Ozcan, Professor Verwesen, and graduate student Hongda Wang, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we didn't have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. These webcasts can be viewed on demand. LabRoots will alert you through email when it's available for replay. We encourage you to share that email with your colleagues we may have missed today's live event. Until next time, goodbye.